Hello, my name is Ken Attie, and I'm the Vice President of Medical Research at Acceleron Pharma. And I would like to thank the FSHD Society for inviting us to present the results from a phase 2 study of ACE-083, which is a locally acting myostatin inhibitor in FSHD. The study recently completed, and we'll be presenting the results and some lessons learned from the study. I'd also like to acknowledge Dr. Statland, who was our coordinating investigator for the study and will be available for the Q&A session. Of course, we also want to thank all the other investigators and their staff, and most of all the patients and their families who participated in the trial. My disclosures are here. I'm an employee of Acceleron and Dr. Statland's as well. So myostatin inhibitors are a class of drugs that are designed to increase muscle mass, and in so doing could help in a variety of disorders. They've been tried in neuromuscular diseases, in age-related wasting, and in disease-related wasting conditions. But today we'll address their use in neuromuscular disease. And at Acceleron, we've been working on myostatin inhibitors for that purpose for some 15 years now. And we've had 3 different drugs in clinical trials. We've had 8 clinical trials in a variety of study populations. And what's shown here is the 3 drugs that we did test. ACE-031 and ACE-02494 were systemic, meaning that when we administer those drugs, they could increase muscle mass throughout the body. And that's how most other myostatin inhibitors work. For ACE-083, we designed the drug so that it would work locally. And you can see that we've highlighted the muscles in the upper extremity and in the lower extremity that we targeted with the drug. So you may wonder, why did we do this? The systemically acting drugs tend to increase muscle, but only by a moderate amount, say about 5% increase in muscle mass. Whereas ACE-083, when we inject it locally, it acts locally and has a more concentrated effect. And we've been able to see increases closer to 15% in the muscles that we target. In addition, since there's very little systemic exposure, it's a very safe way to administer these drugs. So in FSHD, we had to select the muscles to target. And you can see here that we selected the tibialis anterior, which I will refer to from here on as the TA muscle, in the lower extremity, and the biceps in the upper extremity. And these were selected because of their importance in terms of activities of daily living and overall quality of life. Obviously, there are other muscles that are affected in FSHD, but these muscles are also more accessible for us to inject ACE-083. The TA muscle is in the front of your lower leg. It helps you to lift your toe or dorsiflex at the ankle. So it's particularly important to reduce the risk of trips and falls. The biceps is in your upper arm, of course. And although it's not one of your shoulder muscles that are affected in FSHD, it is still an important muscle for carrying, for lifting, and also for bringing your hand to your body or to your face for activities of daily living. So how do myostatin inhibitors work? We act by blocking myostatin, also known as GDF8, which is a member of the TGF-beta family of proteins. And these proteins tend to regulate systems throughout the body. And one of the systems that it regulates is muscle growth. And so myostatin, along with other proteins like activin, will bind to receptors on muscle cells as shown here. And when it does that, it actually inhibits muscle growth. So you may wonder, why would we have something like that? And the reason is that we have a number of other hormones like growth hormone and testosterone that stimulate muscle. And so this is sort of a system that puts a brake on that, and it inhibits muscle growth. And if this system is overactive, you may not get enough muscle growth when you want it. So what we've done is we've designed a drug that would bind to myostatin and activin and prevent it from interacting with the receptor. And in that way, we enhance muscle growth. And we happened to take advantage of a natural ligand trap, if you will, called follistatin, that the body produces for the purpose of regulating myostatin and activin, and we created our drug based on that. The study that we performed was in two parts. Part one was a dose ranging study, and part two was the double-blind placebo-controlled part of the study. So in the dose ranging part of the study, we treated approximately 18 patients in the TA and another 18 in the biceps. And we did it at 3 different dose levels. So we did 150, 200, and 240 milligrams per muscle. And sometimes we treated unilaterally, and then later we tried bilaterally. So the purpose of this part of the trial was to see if the drug could be administered and if it was safe and well tolerated, and then if we were getting the muscle growth that we wanted, and finally to select which dose to take into part 2. And so the study was successful in increasing muscle, and we found that 240 milligrams was safe, and it produced the largest number of patients responding with that 10 to 15% increase in muscle that we were looking for. In part 2, we had patients divided into the active treatment group with ACE-083 and the placebo-treated group. So half the patients received active drug and half the patients placebo for 6 months. After 6 months, everyone received ACE-083. So we call that the open label part of the study. The drug's administered by direct injection into the muscle that we're trying to treat. And typically it was 4 injections, and it was given every 3 weeks. We recruited adults with FSHD, and they could have FSHD1 or FSHD2. This kind of treatment, since it's targeting muscle, it really doesn't matter what the genetic component is. Primary endpoint of this study was muscle volume, as measured by MRI, and we used a technique that looked at the entire TA or biceps muscle and calculated the volume of the muscle. So you would ask, why is that the primary endpoint? The reason is that muscle growth is critical to how ACE-083 would work, and then we would hope to see improvements in strength, function, quality of life, if that happened. We were not sure which functional measure we were going to choose as a phase 3 endpoint in order to get approval. So at this point, we used muscle volume as the primary endpoint. But we had all the others as secondary endpoints. And so this included measuring the fat fraction in the muscle. For example, how much of the muscle was replaced with fatty infiltration. We had performance tests of the upper limb as well as the walking and climbing tests for the lower limb. We had a patient-reported outcome measure for quality of life known as the FSHD Health Index, and we had strength measurements. So who were the patients that we recruited? I'll show you the data for part 2, but it was very similar for part 1. And basically, these patients had a median age of around 45. You could see that in the TA group, it was about 50-50 male-female, but we had somewhat more males than females in the biceps group. There was a significant amount of fat replacement of the muscle, upwards of 20-some%, but that varied greatly from one patient to another. Most of the patients had FSHD1, as you might expect. And about 10 of the patients had a severe contraction of the D4Z4 fragment to 1 to 3 repeats. Patients had a wide range of duration of symptoms prior to entering the study. The median was around 20 years. They had mild to moderate weakness, either in dorsiflexion of the ankle or elbow flexion. And the muscle mass was decreased compared to what would be expected for these subjects. So I'll start with the imaging results, since that was the primary endpoint, and that is results of the MRI tests. And what we found was there was a significant increase in muscle volume as we expected compared to placebo, and that ACE-083 caused about a 16% increase in total muscle volume in the biceps group when you took the mean for the ACE-083 group and subtracted the mean for the placebo group. And in the TA, that was about 9.5%. And you can see in green here which ones are statistically significant. Now, when we just looked at the contractile muscle, so if we took away the fatty infiltration, you're left with that viable contractile muscle, the increases were closer to around 20% compared to placebo. And that increase in the contractile muscle component caused there to be relatively less fat, and that's the fat fraction, or the percentage of muscle that was fat. And that decreased, though not significantly, in the biceps group and more significantly in the TA group. What about strength and function and quality of life, or patient-reported outcomes? I'm going to start with strength in that we measured strength a couple of ways, but the techniques for measuring strength, for example, with a handheld dynamometer, are not ideal, and they have a high amount of variability. So the study was actually quite underpowered for measuring strength, and we don't believe that strength could ever have been a phase 3 approvable endpoint. We saw a transient improvement in strength in the biceps group that was not maintained, and we did not see an improvement in strength in the TA group. However, we would have needed hundreds more patients for this to be a viable endpoint for study. So I'll concentrate on the functional tests and the quality of life tests. And you can see them here. This is the results after 6 months. So we're just looking at the placebo-controlled period. First column are the changes in the placebo group, and the second column, the changes in the ACE-083 group. And the third column is the difference. So we subtract ACE-083 minus placebo. And you can see none of these are green in the sense that none of them were really statistically significantly different. There was an improvement of a few percent in the performance of the upper limb, looking at the middle level domain of that test in the biceps group, but it was in the range of around 3%. For the FSHD high, we did not see improvements. An improvement in FSHD high score would be a decrease in the score. And we're looking for decreases in the range of maybe 8 to 10 points to be clinically meaningful. We did see an improvement in the shoulder arm score in the biceps, but it was more in the range of 4 to 5 points. In the TA group, we did not see improvements in the 6-minute walk test, the 10-meter walk run, or the 4-stair climb, as well as the FSHD high. So look at some of these in the graph form. If you look at these tests in the TA group, we have the 6-minute walk test where we're looking to improve or increase the distance that you walk in 6 minutes, and the 10-meter walk run where we're trying to decrease the time it takes you to walk or run 10 meters. And so that percentage change from baseline is shown on the y-axis and the study day on the x-axis. And up until the dotted line here, the red is the group on ACE-083 and the tan is the group on placebo. After the dotted line, the placebo group switches to ACE-083, as I mentioned, and gets treated for 6 months with active drug. So you can see a few things in the 6-minute walk test. One is that the ACE-083 group for the 12 months did not have an improvement in 6-minute walk test distance. And the same is true for the placebo group, even when they switched to active drug here at the dotted line. What is apparent is that for the first 2 or 3 and maybe even 4 measurements of the 6-minute walk test, there was an improvement even before we started treatment during the screening period. And that occurred in both the placebo and the treated group. So what this represents is what we call a learning effect. So the patient is learning how to do this, the test better with repetition. And we've seen that many times before. So there's this increase, which is pretty significant, that has nothing to do with the treatment. It actually occurs before treatment. In the 10-meter walk run, we saw no change in the active group, and we saw this initial decrease, but then no change in the placebo group, and it did not decrease when the placebo patients switched to active drug. So what about the biceps group? So in the biceps group, we saw that small change in the performance of the upper limb. But perhaps more interesting was what happened with the patient-reported outcomes. So patients answered over 100 questions in the FSHD Health Index, and that total score is shown on the left. And here we're looking for a decrease, as I mentioned, of maybe 8 or 10 points. And unfortunately, we did not see that decrease in either the placebo group or the treated group. There was a transient time point here where there was a few points decrease in the treated group in the second half of the study. However, if we looked at one of the subscale scores, so a few of the questions related just to the shoulder and arm, we could see slight differences between the two groups, with the blue being ACE-083 and the tan being placebo here at 6 months that I mentioned before. There's a few points. However, as the study continued, that decreased further in the active treated group, and the placebo group was pretty flat. Now, unfortunately, we did not have a placebo group to compare this to in the second 6 months. We don't know if something like this might have happened with 12 months of treatment, even in the placebo group. In addition, when the placebo patients switched to ACE-083 after 6 months, they had no improvement in the shoulder arm score over the next 6 months. With respect to safety, as I mentioned before, this drug has very little in the way of systemic exposure. So most of the adverse events that we saw were related to the injection site area. So you see injection site redness, pain, warmth, and as well as some myalgia in the muscle that was being injected. But otherwise, this drug was very well tolerated in both the treated and placebo group. Some adverse events such as muscle pain, myalgia, was more frequent in the ACE-083 group than in the placebo group. We had no drug-related serious adverse events. So in conclusion, in this placebo-controlled part 2 of our study of ACE-083 and FSHD, we did see a statistically significant change in muscle volume, a significant increase in both the TA and biceps muscle in the ACE-083 group versus placebo. And this caused there to be a relative decrease in fat fraction in the TA group. However, these changes that we saw in the muscle were not statistically significant in terms of their effects on either the functional endpoints, such as the motor function test, or the quality of life patient-reported outcome measure. And that's true for either the TA or the biceps group. We did show a trend for an improvement in the FSHD high shoulder arm subscale score in the biceps group. But this was observed mostly in the second 6 months when we didn't have a placebo control to compare it to. So what did we learn from this study? We learned how to conduct trials effectively, how to train sites to do the various parameters that we measured. And we also noticed that it was fortunate that we had a screening period for some of our parameters because there was a significant learning effect during that screening period, especially for the 6-minute walk test, which tends to improve as you learn how to do the test. So our recommendation for future FSHD trials is to consider having this run-in period, especially if you're going to use something like 6-minute walk, so that you can reach the same baseline in the treated and placebo groups. Which brings us to the placebo group. The placebo group is very useful for interpreting the results, both for things like the walking test as well as questionnaires where you're asking patients how they feel. We didn't have a placebo in part 1, which meant that some of the conclusions we had from part 1 didn't keep us from going into part 2, which then turned out did not show significant differences when compared to placebo. So I think these are our recommendations. I will point out that for the FSHD high, we did not see this learning effect. So that's a good outcome in terms of using this patient-reported outcome measure in the future. So with that, I'd like to just turn to a quick summary of some of the other myostatin inhibitors that have been in development for neuromuscular diseases. It's a long list here. I think most of you may have heard about some of these companies and their drugs and the different indications that they've been tried in. You can see that we're not alone in having some of these products be discontinued, either due to safety findings or efficacy lack of, and that the only 2 that are active on this list are now being tested in SMA. So maybe this new disease target population will fare differently, but the drugs are relatively similar to the ones we've used before. So with that, I'd like to really thank all the different sites that participated in this trial. This study was conducted in the US, Canada, and Spain. You could see the principal investigators here, large number of sub-investigators, the physical therapists, and the study coordinators really did a terrific job conducting the study. But most of all, I'd like to thank, I'd also like to thank MedPace, which is a CRO that helped conduct the trial, and our friends at Acceleron. But I'd really like to take a moment to thank the patients and their families and their caregivers. This was not an easy trial to do, coming in every 3 weeks for injections, but they adhered to the protocol really, really well. It was terrific in terms of that. The company was certainly grateful that we had a very clear and outcome from the study so that we could make decisions on how to proceed. Just to reiterate, Acceleron has decided to discontinue development of ACE-083. We had similar results in another neuromuscular disease called Charcot-Marie Tooth disease. And so with these two studies results, we decided to discontinue development of ACE-083. So with that, again, I'd like to thank all of the participants in this trial, and thank you to the FSHD Society.